So, hello and welcome to the talk with the title A Switch in laminin beta 2 to laminin beta 1 isoforms during aging controls endothelial cell functions and enhances endothelial-to-mesenchymal transition. My name is Julian Wagner, and currently I'm working in the Institute of Cardiovascular Regeneration of the Goethe University in Frankfurt under the supervision of Professor Stephanie Dimmler, where I also promote at the moment. And I'm also an active member of the German Center for Cardiovascular Research and obtained my training in Gießen in biochemistry and also physiology. So first of all, I'm going to give you a brief introduction to my topic. Endothelial cells are the inner lining of blood vessels. They are very important to maintain oxygen and also metabolite transport towards the tissues and maintain vascular integrity by interacting with different other cell types, such like smooth muscle cells or parasites. Besides this, endothelial cells also play a role in maintaining tissue homeostasis and control tissue regeneration in a paracrine fashion by producing paracrine factors and also matrix proteins. So by changing the microenvironment or the vascular niche, endothelial cells has been shown to drive regeneration in lung, bone marrow, and also in liver. However, with aging, we see a decrease of these regenerative capacities and also an impairment of angiogenesis. So the aim of my study is to find out or to decipher how the endothelial cell niche is regulated with aging. To start with the project, we first isolated lung, liver, and heart tissues from young and aged mice, isolated then the perfused endothelial cells, and performed RNA deep sequencing to see which genes were up or downregulated. Since we are interested in the vascular niche, we first focused on matrix proteins. In cardiac endothelial cells, we found a dysregulation of the laminin beta chain. With aging, we saw an increase of beta 1 laminin, whereas beta 2 laminin was decreased. To confirm our data, we performed immunohistochemistry. You can appreciate on the left side immunostainings of beta 2 laminin in young heart sections and immunostainings of laminin beta 2 in aged heart sections on the right side. You can appreciate a decrease in beta 2 laminin expressions with aging. To confirm that we really saw endothelial matrix proteins, we also performed an isolectin B4 staining to stain against endothelial cells. And indeed, we found a co-localization of beta 2 laminin and also endothelial cells below. For beta 1 laminin, actually, the case wasn't that clear. So we could find a certain increase in beta 1 laminin, but this one wasn't really significant. But we could observe a significant increase in endothelial cell and laminin beta 1 co-localization with aging. Besides this, surprisingly also in acute myocardial infarctions, we found the same phenomenon. So upon AMI, so acute myocardial infarctions, endothelial cells start to increase beta 1 laminin expression and reduce beta 2 laminin expressions. We also confirmed these data by using immunostainings, and you can see on the left side the infarcted zone of a heart, and here's mostly beta 1 laminin expressed, which you can see in red stainings. On the right side, you can see the remote zone where mostly beta 2 laminin is expressed. So we're very sure that this change in laminin is very important or very crucial for the organism. So we start thinking about laminin. So laminin is a trimer consisting of 1 alpha, 1 beta, and 1 gamma chain. Currently, there are 5 different alpha, 3 different beta, and 3 different gamma chains known, yielding in up to 15 different laminin isoforms. The expression of these isoforms differs among different tissues and also among different developmental stages. The nomenclature of laminin is pretty simple. So the laminin 111, for instance, contains the alpha 1, beta 1, and gamma 1 chain. In our DeepSec data, we found that young cardiac endothelial cells mostly expressed laminin 421, where aged endothelial cells mostly expressed laminin 411. To start characterizing the function of laminin on endothelial cells, we performed in vitro assays. First, we cultured UVECs, so human venous endothelial cells, on cell culture dishes that were coated first with laminin 421 to resemble the young matrix, and laminin 411 to resemble the old matrix. What we found out first was that the number of cells was significantly reduced in the presence of laminin 411, although we seeded the same amount of cells in the beginning. We were able to exclude apoptosis and also cell proliferation for being the reason for this, but we found that the cell adhesion towards laminin 411 was significantly reduced compared to laminin 421 or fibronectin, which was used as a positive control. We went further and performed a migration assay that was based on Boyden chambers. In the beginning, we first coated the filter of these Boyden chambers with the laminin 421 or 411 and fibronectin, seeded then again UVEX, and let them migrate for 3 hours. After this, we counted the cells that migrated through this filter. You can appreciate that the migration in the presence of laminin 411 was also significantly reduced compared to laminin 421 or also fibronectin. As a last functional assay, we performed tube formation in the presence of laminin 411, 421, or only metrical. In line with our first findings, not only the tube length was significantly reduced, but also the number of branch points, so the degree of network formation was significantly reduced in the presence of beta-1 laminin compared to beta-2 laminin. So what we found so far was that with aging we have a change in matrix proteins. But what happens to the parent factors that were released by the endothelium? To characterize this or to answer these questions, we again seeded UVEX on both laminin isoforms, kept in for 24 hours under basal medium, supplemented only with 0.5% BSA, collected the supernatants, and performed a dot plot to determine the secretome profile of both cultures. You can appreciate in this graph the secretome profile of the laminin 421 culture in white and of the laminin 411 culture in black. What you can see is that different proteins like thrombospondin 2, PTGF, FGF basic were downregulated in the presence of beta-1 laminin, but other proteins like VEGF-C were upregulated. But what does it mean? So FGF basic or FGF2 is known to inhibit endothelial-to-mesenchymal transition in the TGF-beta pathway. TIM1 is an endo-MT marker, and VEGF-C was shown to induce epithelial-to-mesenchymal transition, so a pathway which is closely linked or related to endo-MT. So endothelial-to-mesenchymal transition, or endo-MT, is a phenomenon where endothelial cells lose their phenotype and start to transdifferentiate into mesenchymal cells, which is driven by different stimuli like TGF-beta, ET1, or hypoxia. In this process, endothelial features such like V-cadherin get lost, but factors like TIM1, alpha smooth muscle actin, and so on, so mesenchymal features, are acquired. So we first checked our deep sequencing data set from the beginning to screen them for endo-MT markers. Indeed, we found a certain upregulation of different endo-MT markers like Notch, SMART, or TGF-beta, but we found a very strong and significant increase of SM22 alpha, so a very prominent endo-MT marker, with aging in the endothelium of the heart. So to investigate now the role of laminin in endo-MT, we again cultured UVEX in both the laminin matrices and the presence of TGF-beta2 to induce endo-MT. As a readout, we performed a staining against V-cadherin, which you can see in green, and against SM22 alpha, which you can see in red. So on the left-hand side, you can see the culture in the presence of laminin 421. There you can see a very prominent and nice staining of V-cadherin in green. These cells show a very characteristic capstone-like structure, which is very typical for endothelial cells. SM22 alpha is only poorly expressed. On the right-hand side, you can see the culture in the presence of laminin 411. Here you see that V-cadherin is only poorly expressed and starts to vanish. The cells are getting bigger and lose their cobblestone-like phenotype, and they start also to express SM22 alpha. So here we see a transition from endothelial cells to mesenchymal cells. But what does it mean, or what is the molecular mechanism behind all this? So from the literature, we know that integrins are the most important laminin receptors, especially the integrin beta-1 subunit. Upon laminin binding, integrins get activated and undergo a conformational change. This change can be determined or can be visualized by using a primary and also a secondary antibody, which is coupled to a fluorophore. And this is exactly what we did. So again, we cut UVEX on both laminin matrices and on fibronectin as a control matrix, and determined the beta-1 integrin activation after 24 hours. You can nicely see that the activation of beta-1 integrin was significantly less in the presence of laminin 421 compared to laminin 421 or fibronectin. So we went further and performed a integrin beta-1 knockdown in UVEX and repeated the cell matrix adhesion assay from the beginning. You can see in white the adhesion towards laminin 421 and in black the adhesion towards laminin 411. Again, we saw that the matrix adhesion towards laminin 411 was reduced compared to laminin 421, but upon beta-1 integrin depletion, we found that the adhesion towards laminin 421 was reduced to the level of laminin 411. Upon beta-1 integrin depletion, the matrix adhesion towards laminin 411 was completely abolished. So we see that integrin beta-1 activity is impaired in the presence of laminin 411. To conclude, we have seen that aging and also acute myocardial infarctions increase the expression of beta-1 laminin and decrease the expression of beta-2 laminin, which affects the integrin beta-1 activity. As a functional readout, we see a drop in cell matrix adhesion, migration and tube formation, but an increase in endo-MT, which leads to an overall impairment of the vascular niche. If you want to learn more about my research, you will find the corresponding paper in PubMed, which we luckily published this year in March in the journal ATBB. And if you want to learn more about myself, you will find a link to my LinkedIn profile on the left corner. Last but not least, I want to thank all of my colleagues in Frankfurt. They helped me a lot in the lab. And of course, I want to thank all my collaboration partners in Münster and Bad Nauheim. And I want to thank you all for your kind attention. Thank you very much.